Good afternoon. Very warm welcome to everybody, particularly to the college pupils who've come from a number of schools around the country, and I hope that you'll all be inspired by the lecture you're about to hear. I also want to welcome other members of the general public. This is a general lecture, so hope everybody from the general public is welcome. In particular, I want to welcome all the attendees at the Neuroscience Ireland Conference, which is ongoing today and tomorrow, which has coincided nicely with this lecture. And of course, also we want to welcome all our colleagues from DCU. I think it's really a great privilege and honor for us to be able to listen to a Nobel Prize winner of 2013 in medicine or physiology. And it's particularly inspiring for ourselves because it's in the area of neuroscience, which has certainly entertained me for most of my career. And I think the award of the prize to Professor Sudhof has really raised the profile of neuroscience in Ireland and has made the funding agencies much more aware of the importance of supporting research in this area. So without further ado, I want to invite the President of DCU, Professor Breen McCrath, who will introduce the speaker. Breen.
Thank you, Oliver. Ambassadori, Ian Agradamula, agus Agina Yushla Galer, ach gohortha na Micléan atá os Maccormac an seo. Tá fáitilóh Galer an seo Hranóna hí Gulscoil Catharváile á Clia don Ócáid John Táchtach seo. Your Excellencies, distinguished guests, ladies and gentlemen, but especially the students who have joined us here today, you're all very welcome here to DCU for this very important event. Also colleagues from other institutions, Dr. Mark Ferguson, Director General of Science Foundation Ireland, all very, very welcome here today. In welcoming the students, I want to highlight in particular the secondary schools in attendance. Boris O'Kane Community College from County Tipperary, Good Counsel New Ross from County Wexford, and Jesus and Mary College in Goatstown, County Dublin. You're all very, very welcome with your teachers. We also have students here from around the world, especially those on the Science Without Borders program from Brazil, but I know we have students from India and China and so from many countries here in the audience. And as Oliver said, I want to offer a particular welcome to attendees at the Neuroscience Ireland 2015 conference, which we are delighted to host here at DCU over the next few days and who are co-hosting this event with us. Just before I introduce our eminent speaker, let me give you some background to the event. Almost 120 years ago, in November 1895, Alfred Nobel signed his last will and testament, giving the largest share of his fortune to a series of prizes, the Nobel Prizes. And since 1901, the Nobel Prize has been awarded to men and women from all corners of the globe for outstanding achievements in physics, chemistry, physiology or medicine, literature and peace. And then in 1968, economics was added to the list of these fields. And it's interesting to look at some of the facts around these numbers. Since 1901, almost 890 individuals and 25 organizations have been awarded the Nobel Prize. But strangely, and I think worryingly, out of the 889 prizes awarded to individuals, only 47 have been awarded to women. And of those 47, only 20 are alive at the moment. So it's a very small number of female Nobel Prize winners. It's interesting that the youngest Nobel laureate, Malala Yousafzai, who was just 17 years old when she received the Nobel Prize for Peace in 2014, and she just did very well in her GCSE examinations, but it's interesting to see someone so young getting the award. Only 10 Irish people from the North and South have received the Nobel Prize. 5 for peace, 4 for literature, and 1 for physics, and that was Professor E.T.S. Walton, who received an honorary doctorate here a number of years ago. So we're hoping that this series, which we've instituted primarily to engage with young people, will stimulate an increase in those numbers for Ireland. So perhaps there's one of the future Nobel laureates for Ireland in the audience here today. DCU and Magnet Networks have been working together for the past 4 years to bring Nobel laureates to DCU here in the Helix. And this is the 6th in the series. And our objectives are very clear for this series. The focus, as I said, is primarily on students. Our aim is to expose students to excellence of thought, excellence of intellect, and excellence of action, with a view to raising their own aspirations, to setting the bar high as possible for their own personal ambitions. There's no reason why one of you could not be a future Nobel Prize winner. And as we will see again today, Nobel laureates are amazing role models of excellence in clarity of thought, depth of insight, and an application of knowledge. So let me, before I introduce today's guest, Professor Sudhof, I just want to introduce briefly to you Mr. Mark Kellett, who is the CEO and founder of Magnet Networks, who's the major sponsor of this event.
Ladies and gentlemen, distinguished guests and students, I'm honoured on behalf of Magnet to welcome you to this morning's lecture. And I think it's 4 years, nearly 5 years, I think, since Brian envisioned the idea of the Nobel Lecture Series. I had a strong passion for finding a way of how we could encourage the mind, encourage the heart of students. I grew up 600 metres from DCU, actually before DCU even existed, as I recall, down St. Pappin's Road, right across here. And neither my family or friends or peers had the ambition or the aspiration, because we weren't surrounded by that aspiration, to attend university. And I ultimately took the long, hard way of studying at night, studying overseas, distance learning, to gain my qualifications. And I only wish at that time I had had the opportunity to participate and engage with events like this, to encourage my own mind and my friends' minds, to push myself, whether it's through vocational training or further academic training. So when the opportunity arose to engage with DCU, with Brian's vision for the University of Enterprise, it made perfect sense for the business Magnet that I run to engage with this event and to provide an opportunity, particularly for the students here today, to listen from, to learn from world-renowned academics, Nobel Prize winners. And I think over the years we've had some interesting titles for lectures: climate change, we've had Schrodinger's cat, we've had, what was the other one? Time, Einstein, and all things cool in the universe. Pretty amazing topics that I never felt 600 meters away up there that I'd be talking about in 2015. So there's two things, little takeaways that I'd say to particularly the student body and lessons I've learned from these events and in talking with the Nobel Prize winners. One is humility, and the other is to enjoy doing what you're doing. And it was Dr. William Phillips who said to me, as I stood waiting for one of these lectures some years back, said, "Mark, the more I learn, the more I realize, the less I know." And that kind of puts things in when people say, I know everything. None of us know everything. Life is a continual journey of educating, learning, and change. And I just say today, enjoy the lecture. Again, we're delighted to participate, and thanks again to the team at DCU for holding these events, and we look forward to more of them in the future. So thank you and enjoy.
Thank you, Mark. Just to give a few words of introduction to Professor Südhof, he holds the position of Avram Goldstein Professor of Molecular and Cellular Physiology at Stanford University School of Medicine. In addition, he's been an investigator at the Howard Hughes Medical Institute, also at Stanford. The brain really is recognized as the great scientific frontier for this time, for this century. And Professor Südhof's research has examined the fundamental chemistry or biochemistry that underlies all brain activity, from consciousness to memory to sensory perception and to movement. More specifically, his research has probed the intricate workings of synapses. These are the all-important junctions in the nervous system, where information in the form of chemical messengers called neurotransmitters get passed from one nerve cell to another, the nerve cell being the neuron. These synapses are fascinating microcomputer information processing units of the brain. In particular, he has focused on how synapses become abnormal during disorders such as Parkinson's disease, autism, and schizophrenia. And in October 2013, less than 2 years ago, Thomas Sudhof was jointly awarded the Nobel Prize in Medicine or Physiology with Jim Rothman and Randy Schekman for their discoveries of machinery regulating vesicle traffic, the major transport system in our cells. So I'm delighted this afternoon, that Professor Sudhof has honoured us with his presence here today and will deliver his lecture to this audience of nearly 500 people. We're also recording the lecture so that it can be viewed online by many students around the country and internationally. So without further ado, I want to invite Professor Sudhof now to deliver his lecture, Molecular Mechanisms of Neurotransmitter Disease.
Good afternoon. It's a pleasure for me to be here. I hope it's a pleasure for you too. I am really, I have to say, in many ways moved by the fact that I'm facing such a diverse audience here today. Nobel Prizes are not accidents for sure, but I don't think anybody ever deserves one. Maybe some, but certainly I didn't, and I still don't. So it is always, in many ways, given, not just for a particular person's work, but for a field, for an understanding, if you want, that has advanced over the last years. And what I'm going to try to do today is to try to illustrate to you why we believe that this particular field that was honored in my case deserves recognition, why it's important. And because this is a very mixed audience, I will try to sort of switch between more specialized descriptions and more general references. If I do lose you because it becomes too specialized, don't despair. I'll come back to more general frameworks as we move on. So today's talk will describe our work over the last decades that accounts plausibly for the mechanisms underlying neurotransmitter release. Some of you may never have heard of neurotransmitter release. Why is it important and why would we want to study it? Well, this goes back to how the brain works. We actually don't know how the brain works, let's be honest. But we do all think that the way the brain in principle sort of works is by communication between neurons, nerve cells in the brain, that continuously and always talk to each other. And they talk to each other at specialized junctions that are called synapses, which are connections between two nerve cells in the brain. And what you see here in this slide on the right is an electron micrograph of a synapse illustrating what it looks like. A synapse is a communication device, but it's more than that. It's also a computational device. And on the presynaptic side, the neuron that sends the signal, that speaks, basically communicates by secreting a chemical neurotransmitter that is then recognized on the postsynaptic side. You will see that on the presynaptic side, there are these vesicles, which are synaptic vesicles, that are filled with this chemical neurotransmitter. And the way this works is that this chemical neurotransmitter that is contained within the synaptic vesicles is secreted when these vesicles fuse with the presynaptic plasma membrane at the active zone to bind to postsynaptic receptors. The principal way how synapses work was discovered about 50 years ago by Bernard Katz, who received a Nobel Prize for this discovery, and that I just outlined to you. Many synapses come in various shapes and forms. They have very different properties, which is very important for understanding how neural circuits work. But despite all these differences, all synapses operate by the same principle. And this principle consists of 3 steps. The first step is that an action potential is generated in a neuron, and this action potential travels down a nerve axon into the nerve terminal, depolarizes it, opens calcium channels, and then calcium channels allow the influx of calcium, and this calcium then triggers the fusion of the neurotransmitters. Afterwards, the neurotransmitters diffuse across the synaptic cleft and bind to postsynaptic receptors to elicit an electrical signal on the postsynaptic side. From this description, I hope it's clear that neurotransmitter release is the key event in this communication process. It is really what initiates the communication from the presynaptic side to the postsynaptic side. And the way how we normally monitor this process, synaptic transmission, is primarily electrophysiologically. What we do specifically is to stick an electrode into a postsynaptic cell and to measure the electrical signals that are produced when a presynaptically released neurotransmitter binds to a postsynaptic receptor and thereby induces an electrical signal. And you can see this as an example here, down here, where you can see these little blips which correspond to fusion events of synaptic vesicles. There are many interesting things about synaptic transmission, but what I always found the most interesting is how fast it is. It is incredibly rapid. It only takes a few milliseconds total. And neurotransmitter release, as the key step, may happen in less than a millisecond, in a few hundred microseconds. And there's three basic processes that underlie neurotransmitter release. One is that the vesicles need to fuse. The second one is that calcium needs to control this fusion. It needs to trigger the fusion. And it does so, as I mentioned already, very, very rapidly in a cooperative manner. And the third process is that the calcium needs to flow into the nerve terminal at a very specific position, right next to where the vesicles actually fuse, right here. So the calcium channels that allow this influx must be localized right here. And these three processes are the ones that underlie the release reaction and initiate the synaptic communication of information. When I started working on this 3 decades ago, almost 3 decades ago, this process fascinated me because it's so important, but also because of its incredible speed and precision. All of our brain function depends on synaptic transmission. And synaptic transmission, in turn, depends on neurotransmitter release. And so I was interested in understanding how is it possible for a nerve terminal to trigger the fusion of synaptic vesicles with a presynaptic plasma membrane in such a rapid manner and in such a tightly controlled manner that is governed by calcium. And so this is basically what my work has been about, that was honored by the Nobel Prize. I think that this work has established a molecular framework that plausibly explains how this exactly works, although there's many questions to be answered. What you see in this slide is a schematic description of this framework, of what it actually looks like. And basically what you see here in hopefully beautiful colors is molecules that are supposed to be proteins, and that are the molecules that mediate the key events that I just described. The synaptic vesicle is schematically shown on the top left here. This is the presynaptic plasma membrane. The synaptic cleft would be here, and the postsynaptic side would be down here. This is the cytosol. So this is a zoom in into a synapse where the vesicle is fusing. And the machinery that mediates neurotransmitter release consists of 3 components, principal components that correspond to the 3 processes, to the 3 basic events that underlie neurotransmitter. A fusion machinery, a calcium triggering machinery, and a calcium channel tethering machinery. And those three machineries, or subsets of the machineries, interact with each other. They work together to achieve the overall reaction of neurotransmitter release. In the following, I will try to briefly summarize the molecular mechanisms of fusion, and then I will discuss what for me has always been the most interesting question, which is how is it possible for an action potential to trigger release so rapidly via calcium? And I will finally touch on our understanding of calcium channel tethering. The three processes that govern neurotransmitter release correspond to these three machineries or three apparatuses that I just illustrated in this very schematic slide. The first one is vesicle fusion. In parallel studies at the synapse and in yeast elucidated the fundamental mechanism of membrane fusion, a fundamental mechanism that governs fusion in many other parts of the cell. Membrane fusion, as it turns out, is an incredibly widespread process that happens in all cells at all times, all eukaryotic cells at all times. And so it's very fundamental not only to neurotransmitter release. It just happens to be highly controlled at the synapse. And because of this control, the synapse, together with yeast genetics, turns out to be one of the key systems where it could be studied. I don't want to go into detail in terms of the membrane fusion reaction because it's been so broadly studied. And so I just want to illustrate to you briefly again how we think it works. And this is shown in this slide here, which shows again the synaptic vesicle in the presynaptic terminal. And this vesicle here fuses with the presynaptic plasma membrane. And this fusion reaction operates in two principal steps. The first step is there needs to be assembly of protein complexes from the vesicle and the plasma membrane in a manner that they form a little device that forces the membranes close together. And this occurs with the formation of so-called snare SM protein complexes that are formed by the snare proteins synaptobrevin, SNAP25, and syntaxin, which assemble into this zipper-like molecule that then forces the membranes close together. The second step is the zippering up, complete zippering up of this complex that causes the fusion port to open, allowing the neurotransmitters to escape at this point. What is interesting about not only this fusion reaction, but all fusion reactions in biology is that because this is a reaction that requires energy, it is supported by enormous conformational changes which actually produce the energy or set free the energy that fuels the reaction. In the case of the synaptic vesicle fusion, there are two fundamental conformational changes that allow this fusion reaction. One is the opening of the snare protein syntaxin and the rearrangement of the SM protein MUNC18 so they can form this complex in the first place, in the first step. The second is the full assembly of the snare complex to force the fusion pore open, which is the second large conformational change involved. These two conformational changes have very different purposes at the synapse, and they are specific to synapses and other calcium-dependent fusion reactions because they allow a better control of the reaction. The close conformation of syntaxin contains this bound SM protein MUNC18 because this, in this state, forms a gate to the actual assembly for the snare complex that then triggers the fusion reaction. The assembly of the snare complexes that forces the fusion pore open, in turn, requires MUNC18 as well in a manner that is not completely understood, but which seems to be essential for the actual fusion reaction. In this reaction, in our view, snares operate simply as mechanical devices that force membranes together, while SM proteins are essential subunits of the snare-mediated fusion reaction. As we begin to realize in biology more and more, it turns out that virtually every protein in some way is associated with diseases. So it's not surprising that even an important process such as this one, that doesn't seem to be able to tolerate any changes, is affected by disease. There's many diseases, in fact, that impact synaptic vesicle exocytosis. What I want to just mention here is one in particular, which is Parkinson's disease. Because it turns out that in Parkinson's disease, there is a gene and protein called synuclein, which is central to Parkinson's disease pathogenesis. And what we found is that alpha-synuclein actually acts as a chaperone, as a helper for the assembly of the snare complexes that fuel the fusion reaction. And that the absence of alpha-synuclein, just as much as too much alpha-synuclein, in fact, cause neurodegeneration because they disturb the assembly of snare complexes. Let me illustrate this a bit with some real data. It turns out that when you completely genetically delete synucleins from a mouse's brain, these mice initially are fine. But as they get older, they experience a severe impairment in their brain function, and severe neurological symptoms. And when you look at snare complex assembly biochemically, as shown here, they have impaired snare complex assembly. At 40 days of age in the top row, yeah, this is dead. At 40 days of age in the top row, this assembly is much less than at 200 days of age in the bottom row, demonstrating that this is a progressive process, just like neurodegeneration in humans, because there's accumulating loss of function. You can reverse this by simply overexpressing synuclein. What you see here in this slide is an experiment where neurons that lack all synucleins were infected with a virus at different concentrations that expresses increasing amounts of synuclein. And as you can see on the right in the summary graphs, there is a direct relationship between the amount of synuclein and the amount of snare complex assembly. So we believe that this is one example whereby a particular gene, in this case a gene that operates as a chaperone, influences this basic process of membrane fusion during synaptic vesicle exocytosis, and impairments of this process lead to neurodegeneration. Let me move on and talk to you about the second process, the one I'm most interested in, calcium triggering of fusion, which is so rapid, precise, and cooperative. At the same time as we were studying synaptic vesicle fusion, we also systematically characterized the proteins of synaptic vesicles, because when I started, my real goal was to actually try to understand how calcium triggers fusion. This process that Bernard Katz had initially identified 50 years ago. And so the idea was that the synaptic vesicle should contain proteins that bind calcium and that must be responsible for the process. And so we systematically characterized synaptic vesicle proteins, and this approach led, among others, to the discovery of synaptotagmin, which we now know is the calcium sensor for neurotransmitter release. What you see here is a schematic view of the synaptic vesicle and its protein components as seen 25 years ago. And what you can see is that there's many colorful proteins, but one of them sticks out, synaptotagmin. When we cloned synaptotagmin, we were fascinated from the very beginning by this protein because of its structure. Its structure is such that it contains a large cytoplasmic sequence that includes two C2 domains, which were domains that had been identified based as conserved sequences. That's where the C comes from, but that had no known function. We found that these C2 domains actually bind calcium, and that the binding of calcium to the C2 domains of synaptotagmin induces lipid binding and snare binding, with the snare proteins being those proteins that are involved in the fusion reaction. And this really immediately piqued our interest, because with those properties, synaptotagmins had at least the requisite properties you would expect for a calcium sensor, for the molecule that senses the calcium coming in during an action potential and translates it into a fusion reaction. However, the C2 domains did not have any of the canonically known calcium binding sequences, and it was unclear whether synaptotagmin really was the calcium sensor. So we set out to try to understand how this protein binds calcium. And we collaborated with structural biologists over the years to get an atomic structure of the synaptotagmin C2 domains. What you see here on top is again a schematic view of synaptotagmin with the two C2 domains binding calcium. And what you see at the bottom is a ribbon diagram that is used by structural biologists to illustrate the conformation of the C2 domains as determined by structural biology. And you can see that they're composed of very stable beta sandwiches made from beta strands with flexible loops at the top and bottom. And it turns out that the calcium, illustrated here as orange spheres, binds exclusively to the top loops and stabilizes these loops. So these studies identified the calcium binding domains of synaptotagmin. Moreover, they allowed us to formulate the architecture of the calcium binding sites as shown illustrated very schematically on the left, which shows that the calcium is coordinated by multiple residues in the loops, top loops of the synaptotagmin C2 domains. In many ways, this was a satisfying result because it eliminated a new class of calcium binding modules that are now known to be widespread. However, it didn't actually tell us what these things were doing. In those days, 25 years ago, knockout technologies had just been developed. It wasn't possible yet to do conditional knockouts, but the ability to mutate a gene in the mouse germline allowed us to introduce mutations of synaptotagmin into the mouse, mutations that would abolish the function of the gene. And when we did this, and we analyzed the mice, we found, gratifyingly for our work, that the knockout of synaptotagmin abolished fast calcium-triggered neurotransmitter release. What you see here are electrophysiological traces. On the left is a wild type, and you have this massive release. On the right is the mutant, and this massive signal that you see here is gone. However, there is still release left, which I will come back to later. So this result demonstrated that synaptotagmin is indeed essential selectively for fast calcium-triggered release. It is, however, not essential for fusion, because you can also trigger fusion artificially in a synapse, as first shown by Bernard Katz, by simply bathing the synapse into high concentrations of sucrose. And when you do that, you get these massive responses as seen here on the right. And they were identical between the knockout and the wild type, demonstrating that synaptotangmin 1 is actually not essential for fusion. It's only essential for calcium triggering. So this work established that synaptotangmin 1 is a synaptic vesicle calcium binding protein and is essential for fast calcium triggered release. Moreover, additional studies demonstrated that synaptotangmin does not act alone, that it needs an accomplice, a small protein that we named complexin because it binds to snare complexes. And this small protein sits right on top of the snare complexes as shown here in this diagram. And the idea is that then calcium acts on the snare complexes by binding to synaptotangmin and moving it onto the snare complexes that mediate the fusion reaction, and that is how it all works. And this shows the crystal structure of the complexin shown here in pink on the snare complex. Complexin is an essential activator of synaptotangmin 1 that is evolutionarily conserved. What I find interesting is that in biology, some processes are present in all animals, sometimes even in all eukaryotic organisms, and others are specialized and only in certain organisms, with the most specialized are the ones that are specific to humans. Among the most primitive animals are the cnidaria, as shown here. What you see here is a cnidarium that is called Nematostella vectensis. It doesn't even have a real brain, it just has specialized neurosecretory cells. But it encodes in its genome synaptotangmins and complexins, whose evolution is contingent with the evolution of animals as such. There's no separation, they're always there in all animals. What we did is we took the complexin from this animal and we used it in a mouse, and actually not in the mouse itself, but in cultured mouse neurons. And when we did this, we found that the Nematostella complexin was fully active in mouse synapses, as illustrated here in the size of the synaptic responses in the absence of complexin. When you add rat complexin, it's rescued. When you add Nematostella complexin, it's rescued too. This experiment demonstrated thus that the fundamental machinery that I'm telling you about is at least in part conserved, that it can actually be exchanged over large evolutionary distances, illustrating that the principle is a very constant one throughout the animal kingdom. So all of this at this point in our studies, most of which was done about 20 years ago, seemed to suggest that synaptotangmin is a calcium sensor for exocytosis. But you know, not only in science, but particularly in science, we all have friends who always doubt everything we say. And so when we proposed this concept, there were a lot of suggestions that were very helpful. Among them, the suggestion that synaptotangmin is not a calcium sensor, but is something of a scaffold that organizes some other calcium sensors. And so to meet that challenge and to really test whether synaptotangmin is the calcium sensor for the release reaction or whether it's some other protein and synaptotangmin just serves as an organizer, as a kind of scaffold, we went back to our description of the calcium binding sites in synaptotangmin and used structure-guided mutagenesis in knock-in mice to explore this. Now let me explain this to you in a bit. I showed you this slide before. This illustrates the architecture on the left, the architecture of the calcium binding site. And because we knew the architecture of the calcium binding site, we knew which amino acids in synaptotangmin had to be changed in order to change the properties of calcium binding. At the time we were doing this, the technology of mouse genetics had advanced, and it was now possible not only to perform knockouts, but to actually change specific sequences within a gene. And we focused on these residues here to design mutations that shift the calcium affinity of synaptotagmin but don't inactivate synaptotagmin as a way of probing its role in calcium triggering of release. What I'm going to tell you about is two amino acids in particular in this entire molecule, one arginine R233 and one aspartate D232 that are adjacent to each other. And what's interesting is that these changes, these amino acids have very different roles. If you mutate D32 to an asparagine, which is very similar to the original amino acid, it turns out, as you can see here in these curves, that it actually binds to snare complexes better than wild type. It's a gain of function mutation. So there's a shift, there's an increase in function. In turn, the R233Q mutation, as shown here in phospholipid binding assays on top, where you measure binding of synaptotagmin to phospholipids, is a loss of function in that there is a lower calcium affinity, an approximately twofold lower calcium affinity for phospholipid binding. So these two surgically introduced mutations have distinct effects on the properties of the molecule of synaptotagmin. They don't block their function, they just shift the function. One increases snare binding, the other one decreases phospholipid binding. And this allowed us to test the role of these biochemical activities in calcium-triggered neurotransmitter release. The D232 mutation that increases calcium-dependent snare binding turns out increased also calcium-triggered neurotransmitter release, as shown here. This is shown here schematically as the amount of synaptic signal that can be elicited as a function of calcium, or on the right as the calcium affinity, which is a higher calcium affinity when the KD goes down. Conversely, the R233Q mutation that decreases the calcium affinity during phospholipid binding decreases calcium-triggered neurotransmitter release and decreases the calcium affinity significantly. So the effect of the in vitro changes translate completely into an in vivo change in calcium triggering, formally proving that calcium binding to synaptotagmin triggers neurotransmitter release. So this established that calcium binding to synaptotagmin triggers fast release. In science, it never ends. You solve one problem, there's a new problem. The next problem that we had was that the genome actually contains more than one synaptotagmin. How could we explain that all this is happening with this one synaptotagmin that we were initially studying when there are so many synaptotagmins? As it turns out, these synaptotagmins are very heterogeneous. In fact, in biology, it turns out that most genes are present in many different similar variants that have different properties. 8 of the 16 synaptotagmins bind calcium and 8 don't. Those that bind calcium fall into two principal classes: blue synaptotagmins that are not covalently dimerized and red synaptotagmins that are. Which synaptotagmin is a calcium sensor? Which synaptotagmins are calcium sensors for fast release? And to address this, we systematically used rescue experiments and actually knockout experiments to test each one of these synaptotagmins. And the way this works is that when you have a synaptotagmin knockout, as I showed you earlier, fast release is gone, as shown here. You then reintroduce different synaptotagmins into these neurons and test whether you can restore release. And indeed, when you do this for synaptotagmin 1, you fully restore release. When you do this for synaptotagmin 2, you also restore release, and for 9. But you don't restore release for any of the others, and particularly not for synaptotagmin 7. So these experiments showed that 3 of the 8 calcium-binding synaptotagmins were calcium sensors for release, whereas the other 5 weren't, including 7 that's very similar to 1, 2, and 9, which was puzzling. It was puzzling because of the similarity. It seemed like 3 of the 4 blue synaptogons are involved in neurotransmitter release. So this created new problems. Why does synaptognon 1 knockout have a phenotype at all if there's 2 other synaptogons that work just as well? And why doesn't synaptognon 7 function in release if it is so similar to the blue synaptogons? To address these issues, we measured in single individual neurons using a specialized technique that is called a fluidigm-based PCR, where you can measure literally hundreds of genes in one cell quantitatively. And what you see here is an array of such measurements for different synaptogons and also other proteins that are expressed in neurons in single cultured neurons shown here. And I don't want you to look at everything. What's important here is synaptogons 1, 2, 7, and 9. And blue means there's no expression, and red means there's a lot. And you can see immediately that SID1 is expressed highly, SID7 is expressed highly, but 2 and 9 are simply not expressed in these neurons that have a phenotype. So this explains why synaptognon 1 knockouts have a phenotype. The other ones are simply not there. They're expressed in other types of neurons, not those neurons. What about synaptognon 7? It's highly expressed. It clearly cannot compensate. What does it do? Why is it so similar? Now, you may recall from the initial results of the synaptognon 1 knockout that there was a loss of neurotransmitter release for the fast component, but there was this little bit of release left that was still calcium dependent. And it turns out that that release can become very important if you stimulate neurons over and over, if they fire a lot of action potentials. So some residual calcium-triggered release remains. And we wondered whether that could be mediated by synaptognon 7. And so we took synaptognon 1 knockout controls and we reintroduced into the synaptognon 1 knockout synaptognon 7 wild type, and nothing happened. It couldn't be rescued. But when we then also decreased synaptognon 7 in the synaptognon 1 knockout with a knockdown, the residual release is gone. And this is also illustrated here. When you reintroduce 7, it's rescued. When you take a mutant of 7, it is not. So these experiments demonstrated that synaptognon 7 is, after all, a calcium sensor for release. It just has different properties. It acts in the same neurons, but it acts sort of in tandem. It's like a tag team, where synaptognon 1, 2, and 9 come first and 7 comes second. It's important because 1 or 2 or 9 cannot always function, and then 7 kicks in. And so all blue synaptogons function in synaptic vesicle fusion, but exhibit different calcium triggering kinetics. They also function actually in other types of calcium triggered exocytosis, as it turns out, hormone secretion, in particular mast cell degranulation and so on. So they are not specific to neurons, but they mediate the fast calcium triggered secretion of all kinds of messenger molecules. Not surprisingly, it turns out that at least some of the red synaptogons are also calcium sensors for other specialized types of calcium-triggered exocytosis, which I won't discuss today. But however, what emerged from all of this is that synaptogons in general are very fundamental calcium transduction molecules that take a calcium signal and transfer it onto the membrane fusion reaction to induce the fusion of specialized vesicles for secretion. So to summarize this is that basically all synaptogons operate by the same fundamental principle in conjunction with complexins to mediate the calcium triggering of fusion reactions. And a recent update, as the story is further developing, is that there's now crystal structures available, not only for synaptogons, but also for the synaptogon snare complex assemblies, and even for the one with complexin. And it turns out that synaptic fragments are an integral component of the whole fusion reaction, not only of the calcium triggering reaction, in a redundant manner, suggesting that their role is more broad than that of a calcium sensor. I would like to now move on to the final brief piece of my presentation, which is the localized calcium influx. Why is it important? Why would it be important to know how calcium gets localized to here and not anywhere else? As it turns out, in the membrane, in this neurotransmitter release reaction that is so specific for neurons, the fusion reaction and the calcium triggering reaction are actually not specific for neurons at all. They happen everywhere in hormone-secreting cells, in any kind of other regulated exocytosis. It is really the localization of calcium channels together with other elements of the fusion machinery specifically to one narrow space that allows neurons and synapses to differ from any other fusion reaction, to differ in speed and regulation. Without localized calcium influx at the active zone, action potentials and release become uncoupled, and release becomes desynchronized and decelerated, and synaptic transmission becomes sluggish, and neural networks won't work. And so the importance of localized calcium influx cannot be overestimated. It's really a key element for understanding the brain. Recent progress in understanding this has allowed us now to formulate a molecular framework for imagining how this might work. And this is more complex than the framework that governs synaptic or the fusion reaction, and is illustrated here. Instead of going through all of these components, molecular components, I just want to show you how one key molecule, a molecule we call RIM, operates in this complex. And I want to use that as an illustration of how we can, at a synapse, actually study the precise coupling of an action potential to a release reaction. This molecule, like several other molecules shown here, is a scaffolding molecule. It basically brings different components together to make them work together. It's an organizing molecule. And we call this RIM because it interacts with the GTPase on the synaptic vesicle surface called RAP3, but it really does much more than that. When you delete all RIM proteins from a nerve terminal, you get a complete loss of neurotransmitter release, as shown here, or nearly complete loss, as shown here, demonstrating that the molecule is important, that this organizational principle of this molecule is important for neurotransmitter release in general. And so this raised the question whether release is impaired because of a defect in calcium influx. One way, which is becoming more and more important in neuroscience to study the brain, is to image it. What you see here is a neuron in culture that is being filled via a pipette which has been used to patch the neuron with a fluorescent dye, and you can see the incredible dendritic arborization of this neuron, the many neurites that this neuron has. You can see that the dendrites have spines, as you can see here in these little protuberances that are all over. As you all know, neurons are characterized by enormous amounts of dendrites and axons. In fact, it's the axon which sends out the signal, which contacts the target. But you'd be hard-pressed to find the axon in this image because axons are tiny. They're very thin. You can hardly see them. And it turns out that the axon is actually this little thing here, which can go on and cover the whole petri dish in which this neuron is cultured. We filled this neuron via the patch pipette, not only with this fluorescent tracer, but also with a fluorescent dye that changes as a function of calcium and can be used to measure calcium. And this allows us to measure both the fluorescent dye that is used to visualize the neuron, here shown in red, and the fluorescent dye that measures calcium, shown here in green, and allows us to measure the calcium signal in a nerve terminal, which is shown here, as a function of an action potential. So with this technology, we could investigate what the role of this particular protein is in the localized calcium influx. We measured calcium in presynaptic terminals. In the wild type, you have this massive calcium signal, as shown here, and this is just another way of plotting the same thing. And this signal is highly dependent on the action potential and happens within a millisecond. When you delete the rims, this signal is attenuated dramatically. And when you reintroduce rim, it's rescued. This is specific for the action, for the presynaptic terminal in the axon, because if you do the same thing over dendrites, there is no change in the calcium signal as a function of an action potential. So these experiments, together with many other experiments, demonstrated that this molecule here is sort of the key element in bringing together the calcium channels to the synaptic vesicle and actually to the fusion machinery. It turns out, in fact, that this whole complex can explain how a nerve terminal operates differently from other fusion reactions, because it organizes into a closed space not only calcium channels and synaptic vesicles, but also the fusion machinery itself. And more recent studies revealed that RIM-BPs, these molecules down here that bind to RIM, are equal partners to RIM in performing these functions. So what I've tried to do today for you is sketch out in maybe too great detail, but I hope you nevertheless got some feeling for the way how this is done in neuroscience, sketch out for you the way how we over the years contributed to an understanding of the molecular mechanisms that underlie neurotransmitter release. I've tried to illustrate to you that there's three fundamental processes that mediate neurotransmitter release: fusion, calcium triggering, and calcium channel tethering. That the fusion reaction depends on a conserved fusion molecules which operate as much in yeast exocytosis as they do in synaptic vesicle exocytosis, but that are controlled by synaptogramins as calcium sensors. Basically, the synaptogramins tell the fusion machinery when to go ahead and open the fusion pore. Both of these elements, as well as the calcium channels that mediate the calcium influx, in turn, are brought into place by a macromolecular complex that serves as a kind of scaffold and binds various components of the machinery to co-localize them. So functionally, the fusion, calcium triggering, and active zone complex molecules form a single interacting nanomachine that mediates fast neurotransmitter release. And I hope that with this understanding, we can now build on this to actually understand how synapses differ, because this is essential for an understanding of brain function. And also, I hope that this can form the basis for understanding how synaptic transmission becomes abnormal when these molecules, for example, are subject to mutation in human disease. In science, we all depend on actually a lot of people to work with us. What you see in this slide are some of the people who contributed to this work over the 3 decades. I won't go into them in detail, but I just want to mention that science is always a team effort. It's never a single person who discovers things. It's always many people. And that I am grateful to all the people over the decades who have worked with me for basically making the discoveries that I in the end get credit for. Thank you very much.
Well done, Thomas. That was very enjoyable. Yeah.
I'm a little slow today.
No, that's fine, yeah. Well, I think we're all impressed with that very lucid and informative description of a topic which is quite complex, but which makes us all tick, as I would say. Without neurotransmitter release, all bodily functions would not operate in the normal way. And never mind the complex things that go on in the brain. Now, Thomas has very kindly agreed to take some questions from the audience. So get your thinking caps on, get your synapses firing. And so we'll start first with some of the school pupils who might have some intriguing questions about these topics. Don't be shy. Perhaps to get the ball rolling, I could ask Tom 1 or 2 questions which have arisen, not scientific questions, but rather on the side. And the first was, Tom qualified in medicine, and I wasn't quite sure what made him switch into pure scientific research. And also, at what stage in his career did he really sort of get the idea of a Nobel Prize in mind? When did he think this was a possibility? I hope that's a fair question.
No, I think it's a very good question. So let me start with the second question, because, you know, as I was saying in the beginning of my presentation, I really don't think that there's anybody, well, maybe some people deserve the Nobel Prize. Yeah, I mean, Albert Einstein, yeah, geniuses. But the fact is, the Nobel Prizes are, although not an accident, are in many ways luck. And I personally, in my career, as it was unfolding, realized that the work we were doing was fundamental and that it was important, and I received many, not enough, especially not enough well endowed, but still many recognitions, and so I knew that I was being considered. But I actually didn't think I would ever win a Nobel Prize, nor did I think about it very much. And the reason is that there's a lot of really good scientists out there. And it's actually very interesting to talk to these people and to see what they're doing. And to this day, I think that it's a question of taste, what you think is the most important at any given time. So I always thought that there were other things that were more important that should get the Nobel Prize, and I still actually think so. And I feel lucky, but it's not something that was really very much on my mind. I knew I was being nominated, but I also know that there's 100 other people who are nominated at any given time. And so there's really no point, I believe, as a scientist to think about these things. I was hoping, in fact, that I would win one of these prizes where they give you a lot of money. You know, the Nobel Prize doesn't really pay that well. But anyway, unfortunately, I won't be able to. I'm no longer eligible for these prizes, which is kind of a pity.
So we have a question in the front. Is this?
No, he's just stretching.
Oh, I see. I thought he had his hand up for a question. Yeah? So let's have a question from the more mature attendees then. Any questions? Don't be shy.
I have a question. Thank you first of all for the fantastic lecture. It's not a science question. I wondered if you could give your own thoughts to some of the younger people in the audience that may be considering a career in science either before they've got to university or maybe some of the PhD students that are here attending the Neuroscience Ireland meeting. What might you say to them if that's not too much to ask of you?
No, it's actually important because I find myself confronted often, especially in the United States, with a certain degree of pessimism about science. And in the United States, you know, objectively that pessimism may be warranted because we live in a country where the majority of the elected representatives of Congress actually believe that evolution is a fraud and that global warming is a fraud. And that is a little depressing, I have to say. But my view is that science in many ways is, if one can say it this way, the soul of our society and our culture, because science depends on the quest for understanding how something works, the quest for knowing what is true and what isn't true about physical phenomena. Okay? And scientists deal with trying to understand really what is there. And I think that people should become scientists who really enjoy figuring out how something works. And it is an incredible time, I believe, to be a scientist despite the political situation, at least in the United States. Not because it's so important, that too, but also because we are in a time where there are so many opportunities to discover things. So the kinds of technological advances that are happening now are really amazing, at least in biology. And just to name a few, so genetics has completely changed how we view and how we analyze life. It has changed it because we now know in human genetics that the human genome is very fluid, that it's plastic, that it changes from generation to generation. That has introduced a tremendous change in the view of certainly neuropsychiatric diseases, because we now know that many of the mutations, maybe most, that predispose certainly to autism and schizophrenia are mutations that happen new, that are not, these are genetic, but they are not inherited, so to speak, if one can call it this. You know, there's a whole world out there. We also know, maybe disappointingly, that knowing the genome sequence doesn't tell you a thing. You actually have to understand what genes do and what it means and what all the other sequences do. We also know now that in the brain, understanding how neurons are connected may be important, but it certainly doesn't tell you how it works. So on the one hand, there are enormous tools now, certainly in biology. On the other hand, there is unbelievable questions that just remain to be discovered and remain to be answered. What I told you about today may be fundamental, I hope it is. I hope they don't take back the Nobel Prize. But you know, it's just a tiny bit. We don't really know how the brain works and what happens when it doesn't work. And so I do hope that some of you guys and girls, in fact, especially the girls, will actually go after this. Another concern that's often said is that there's not enough money and not enough funding and not enough careers and not enough opportunities and so on. And yes, it may be hard, but I don't think it's ever been easy and in fact, at least in the United States, there's actually a lot of money. And much of the money is probably not distributed right, but there's still a lot of jobs. And I think that that should never be a reason not to actually do what one is interested in. Now, having said all this, I do think that for scientists, it's really important to be truly interested in discovering things. Science, in some ways, is dependent on the ability of a person to enjoy doing science. I think that when people do science for other reasons, it's probably not a good idea, because the other rewards in science, like income and money and so on, are not that big. And so that shouldn't be the reason to actually do a scientific career. So that would be the only, I would say, sort of downside. Okay? Except if you win one of these big prizes, but don't. then maybe.
Any more questions? Yeah, here.
Yeah, thank you, Professor. That was an excellent talk and lecture. It was very easy to understand it for a lot of us who wouldn't be experts in this field, indeed. Just a question, with all the advances you mentioned there, molecular biology and indeed your own work there, how far are we away from, if ever, getting cures for diseases of the brain like Alzheimer's and dementia? Thank you.
Sure. Where are you? I can't see you. Oh, over there. Okay. That's another subject that we were briefly touching on earlier today in our private discussions, which I think is incredibly important and which in many ways actually relates to what I just said about the future of a career in science. There are tremendous challenges in human disease. We have no therapies for any of the major brain diseases. And that is amazing because there are so many and they are so important. The reason for that is that we cannot develop therapies against diseases that we don't understand, and in my opinion, we don't understand, for example, Alzheimer's disease. We don't actually know how the disease occurs, why it occurs. We have gene mutations, but we have some really good cell biology. So, having said this, that doesn't mean that we are not close to getting to therapies. In science, maybe the most exciting aspect of it is that you never know how close to the goal you are. It's very different from running a marathon or 100 meters, if you prefer, where you exactly know you have to run that much to get to the end. In science, you don't know if you're running 100 meters or a marathon. You just run, and you hope it's 100 meters. So the fact is that we don't understand any of these diseases, but any of the experiments that people are doing could get us to an understanding that would result in a therapy. So I don't know how close we are. My gut feeling is that for the neurodegenerative field, we are actually quite close, because there has set in a better sense of realism after decades where people were convinced that they understood it with the amyloid cascade hypothesis. I believe that people are now becoming more realistic and see that we need a better understanding of the disease itself, and that there's many labs that are trying to do this understanding. In general, I feel very strongly that in translational neuroscience, and maybe all translational biomedical research, we need to focus on understanding the disease before we can focus on developing drugs. If we don't actually know what we should make drugs against, we can't make drugs. The understanding of the disease has to be priority. I'm saying this because, especially in neuropsychiatric disorders, schizophrenia, Alzheimer's, people have been trying to develop drugs without understanding the disease for decades. And in all of these disorders, there are really either no effective drugs or drugs that have enormous side effects. And so I think that for funding, it is really important to direct the funding first at trying to understand the disease before one tries to actually develop the drugs against the disease, because otherwise it's only guessing and a waste of money.
Okay,
Anybody else?
If there are any more questions? Where?
Good afternoon, and thank you for the lecture. Very much enjoyed it. As a follow-up to the last question, which I think was excellent, and you gave a great answer as to where science is generally trying to deal with these questions. Specifically for your own work and your own research, do you have an imagined application, or do you want to see it going in a particular direction that might lend to one of these therapies in time, or is it too early yet?
I think that my own work is actually very relevant in a way that I just alluded to. So specifically, not by design, it turns out that many of the molecules that we initially discovered are intimately linked to specific diseases. There's a whole area of research that I haven't discussed, which now forms the center of what we do, and which deals with how synapses form and become specified. It turns out that key molecules that we discovered some time ago, called neurexins and neuroligands and other ligands in this whole arena, are mutated in schizophrenia and autism. And so I think that there is a true opportunity to go from the gene mutations to the functional changes to therapeutic intervention. This opportunity, however, depends on understanding what the gene mutations do. In general, the revolution in human genetics over the last decades due to advances in DNA sequencing has been enormously positive, because we know so much more about what happens in disease than we ever did, and about the biology of the genome. However, the one little, I think, caveat here is that we should all be clear that understanding gene sequences requires more than sequencing the gene, and that understanding a disease requires more than knowing mutations in specific genes that cause the disease. And so the opportunity is now there to actually take these mutations, which in my case, in our research, are mutations that are associated with schizophrenia and autism, and to translate understanding these mutations to an understanding of the disorder. And I think that that has a plausible chance of success. How long it's going to take, I don't know.
Okay, I think we've exhausted all the questions. I'd just like to thank once again Thomas for, you can see the enthusiasm he has for his research. I think he's been extremely modest. He's saying, did he really deserve the Nobel Prize? Well, those of you who have any doubts about that should just look up his publication record. And he's published consistently in the very highest impact journals. I won't ask him what his H-index is, but I'm sure it's pretty impressive. So I think there's no doubt about it that his contribution has really been a milestone in the development of neuroscience research, and I'm sure to lead to further developments in the future. So with that, I want to thank everybody for attending, and hope the school children have got motivated. Some of us are too old now, it's a bit late for to be motivated, but you young minds are in the process of development. And one thing I was going to ask Thomas, but if you really want to get into neuroscience research now, because of the interdisciplinary nature of the subject, the question is, what subjects should you study? Probably some physicists down there, there's got to be physics and lasers and so on. Other people would say genetics, other people would say biology and chemistry. I'm a biochemist myself, so that gives you a good mix. So anyway, that's...
You should all study biochemistry for sure. Yeah.
So with that, we'd like to again show our thanks and appreciation for this.
Thank you very much.
It's like being at the Oscars.